Hello, ladies and gentlemen. Mr. Hescox back, starting the next section in your Life Science, Chapter 1. We're going to do the first half of Section 2 on how they explored the parts of the cell, and then we'll start into the cells here later on this week. So the question here is, what is spontaneous generation? Spontaneous generation is the process of life coming from non-living matter. If you would give me an example like meat and flies, rocks for insects, things like that. We're going to look at the types of microscopes and how each of the three main types of microscopes can be used to help scientists study certain things. Tonight's reading that goes along with this is page 18 to 24 in the text. How have microscopes expanded our knowledge of the cell? Cells are measured in very special units called micrometers. Use this Greek letter mu. That's not a U, that's a mu. It means 1/1,000,000th of a meter. If I took a meter stick and chopped it into 1 meter or 1,000,000 pieces, I would have a micrometer. Most cells are small enough that they are anywhere from 1 micrometer to 1,000 micrometers. The light microscope led to the discovery of cells. Now, light microscope uses lenses to magnify the amount of light entering your eye. So I'm going to take a look here at this picture, and we're going to mark it up a little bit and talk about the microscope. So here on your microscope, we have some lenses. This is where your eye goes. We call this the ocular lens. And then we also use these lenses here, which are your objective lenses. And what happens for your light microscope? Light travels up from your light source, is focused through here. This is a 10 power lens, up through and up to your eye. Once it travels up to your eye, it is now magnified. You actually, in this case, most ocular lenses have a 10 power objective. This objective right here has a 10 power. What happens is you take 10 times 10, and everything you look at under that lens will be 100 times bigger. There are two other types of microscopes. We do not have these here. Not everyone is going to get a chance to use one of these in life. You have the TEM, or the transmission electron microscope, which is what this is, or you may have a scanning electron microscope. Both of these use a large vacuum tube. This piece right here has a compressor that pumps out all of the air. There's no air. So if there's no air, obviously nothing's living inside. And then you put your specimens down in here, and the specimens have to be coated in a very fine dust. They used to use gold dust. Today they use silver dust. And when they do it, they actually run it through electricity, and it can magnify the images. So a straight up TEM, they used to say 1,000,000. It is way above that. It's in the 10 million range now. Now, the scanning electron microscopes didn't used to go all the way up there, but they're over 1,000,000 now, depending on the value. The closest electron microscopes to here, Penn State has a lot of them. There's a place called the Millennium Science Center on Penn State that actually has like 15 different types, and they have a really big one that I'm going to talk about here shortly. This is the gentleman who runs it. He is in charge of, this is called the Titan, and it is at Penn State University. I had the opportunity to take kids there for a few years, and this actually, instead of going down on the nano scale, this goes to the pico scale. Now, it's hard to picture this, but it was over $7 million for this machine, and it's like 14 feet high. And the way that this gentleman described it to me is if we could make an atom hold still, we could zoom in and look at it. So let's kind of review here. What type of microscope would you use to view each of the following and why? I'm going to pause this, give you a chance to write your answers out on a piece of paper so we have our thoughts. Then I'll come back and we'll go over them. All right. Did you have a time to think? Let's see what you came up with. So we have an Ebola virus. Now viruses are very, very, very small. So you want the most magnification you can. Most people are going to use a TEM to look at viruses. The reason being huge magnification. If I wanted to look at a moving paramecium, for a moving paramecium to be moving, it has to be alive. The only microscope that allows us to look at living things is a light microscope. And the reason it allows it to be alive is that it doesn't have to coat them with metal or put them in a vacuum. So we're going to say that they're alive. And then if you're looking at the surface of HIV, HIV is the virus that causes the disease AIDS. If you're looking at the surface, you typically are going to make a three-dimensional model, and that's the SEM or scanning electron microscope, and that's because it's 3D. All right, we'll be back on our next lesson to talk about cells, but we had to talk a little bit today on how we find our information about cells. Make sure you get your readings back. Be good and be safe.